Hello students and welcome to another lecture of genetics. In today's lecture, we will be studying about gene mutation and DNA repair. Gene mutation, as you might remember from previous class, is an evolutionary mechanism because that causes changes to the DNA, and those changes to the DNA changes the frequency of the alleles. However, those mutations can also be repaired. Those changes in the DNA can also be repaired, and that's what we are going to cover in this lecture. Just a few reminders. Remember that the DNA is a macromolecule, and it's a macromolecule that stores the information about any organism. This molecule has the property or has the ability to replicate, meaning that one molecule of DNA can replicate itself, producing two molecules of DNA. This molecule is also transmitted from one generation to the other. And that's the whole point of our first classes, to understand how these genes are passed from one generation to the offspring. The DNA is also how the information about the organism is coded. Remember that the DNA goes through a process where it is transcribed into a molecule of RNA, and then the RNA is then translated to protein. And the protein is what makes the organisms. This is one of the macromolecules that helps making the organisms, right? So we can assume that the DNA carries the information to make up this protein. The changes associated to the DNA sequences can give rise to variations, and let's understand these variations as changes in the allele frequency. Or changes to allele frequencies. Okay? When you change the frequencies of those alleles, when you change the genes, you then change the phenotype of those organisms. Remember that the phenotype is how the organism appears physically, right? Therefore, if you change the alleles, you can also change the phenotype. Changes in the phenotype are important because they will allow for adaptation of that organism to the environmental changes that they might be facing. Remember that those environmental changes are causing what we call selective pressures. Okay? Remember that in class number 5, I believe, we used the example of the birds, that when the season was dry, then the birds with a stronger and bigger beak would survive. And then when the seasons were wet with rain, then those with smaller beaks would survive, right? So the changes in the environment, we call them selective pressures. And that selective pressures allows for the evolution of that species. Okay? Also, remember that evolution is changes to the allele frequencies. We've talked about that in class number 5. Those mutations actually cause a change in the base pair of that DNA. So if you think of a DNA as a double strand, right? That DNA has a backbone made up of sugar and phosphate, and then the base pairs, which are represented here. Changing those base pairs is the basis of the mutation. Okay? Now let's look at how those mutations can be classified. We are going to now understand the types of mutation, how those mutations can be classified according to the effects on the function, how the mutations can be classified according to the effects on the phenotype. Okay? Moving on, mutations, like we said, is any alteration in the nucleotide sequence in the genome. Okay? Genome and gene let's use for now as a synonym, but genome is all the genes in that population, in that DNA. Okay? So a mutation is any change in the base pair in the sequence of a gene. That change in the base pair can be a single substitution, meaning that only one base pair was altered, or that can be a multiple base pair substitution, meaning that more than one base pair was changed. Those mutations can also cause a deletion or insertion of the base pairs, and I'm gonna give you an example of that soon, but just think about the molecule of DNA with the bases, nitrogenous bases, right there. Insertion occurs when one new base pair is inserted in that sequence. And as you can see, the sequence now is altered. Deletion means when this base pair is removed from that sequence. And as you can think, inserting or deleting base pairs in that DNA can change not only the gene but also the chromosomal structure. Because remember, the chromosome is carrying the DNA compacted. So if you change those base pairs that make up the DNA, you change the structure of that chromosome. The mutations, they can occur in regions, in specific regions of that DNA. Those regions can be coding or non-coding regions, meaning that a coding region is a region that will generate a protein, and a non-coding region does not generate a protein. It can also happen, mutations can also happen in regulatory sequences, which would be sequences of genes that regulate other genes. Okay, those are sometimes called, or those are called transcription factors. Mutations can also occur with promoters, enhancers, or splicing signals. Now let's look at the type of mutations, because they can be classified according to the molecular change that they cause to the DNA. First of all, we have the point mutation. The point mutation is when one single base pair is changed or substituted to another. A missense mutation is when the alteration in that sequence results in a new triplet code, which results in a different amino acid. So remember that in the DNA, and here I'm just going to represent one strand, you would have a sequence of A, T, G, C, A. And let's say that the sequence A, T, G, C would give the amino acid, let's call that amino acid X. Okay? But now let's say that this base pair was changed. And instead of having a ATC, now you have ATG, now you have a CG. And because of this missense mutation, you have a new triplet code, which causes now the amino acid from being X to be Y. Okay? So missense mutation results in a new triplet code. Triplet means 3 letters in sequence that codes for one specific amino acid. Here, the change in the base pair gives you a different amino acid. A nonsense mutation is when the change in the DNA results in a triplet code for a stop codon. Remember that the stop codons can be UAA, UAG, and I believe it is UGA. Those are stop codons on the mRNA. Here, they do not code, they do not carry any amino acid. That's why they are known as stop codons. In a nonsense mutation, you insert a change in the gene sequence that causes the triplet to look like this, or this, which results in terminating the translation prematurely, or before when it's actually supposed to terminate. We also have a silent mutation, which is when the new triplet code still codes for the same amino acid. So let's say that in our example above, we changed ATG to ACG. And let's say that in this case, you still produce the same X amino acid. Therefore, we would call that mutation silent because there are no changes in the amino acid sequence. If there are no changes in the amino acid sequence, then there are no changes in the protein formation. We also have neutral mutations, which are the mutations or the changes that occur in non-coding regions. Non-coding regions are regions, like the name suggests, that do not give rise to any sort of amino acid or any sort of protein. Okay? There is no gene associated in that region. It is important also that we talk about the frameshift mutations. The frameshift mutations, they result from insertions or deletions of nucleotides in the sequence in that gene sequence. The insertion or deletion of the nucleotide can shift the reading frame. So, thinking about our sequence of DNA here. Okay? Let's say that you have A, T, A, C, and G. Let's say that you have this triplet codon and this triplet codon. But now you entered a new base pair. Let's say that we're talking about an insertion of a nucleotide. And this new base pair is now a G. Can you see that now, and I'm gonna change the color, that my triplet changed. Now my new triplet is ATG, AAC, and the G goes with another triplet, of course. Here, the mutation changed the reading of those codons. Therefore, when you change the reading of that strand, you change the translation, you change the protein that is originated from that reading. Okay? Some of these mutations, like we said, can insert a nitrogenous base or a nucleotide that codes for a stop codon. And here are those stop codons. I have a very good example here of what those mutations would look like. So let's work with the phrase, the cat saw the dog. See that each 3 letters that we have here is a triplet codon. Okay? And let's say that the first triplet would give us the amino acid X, the second triplet would give us the amino acid Y, Z, W, and let's use another letter, B. Now, let's look at the point mutation when we change only one letter. Here in our first example, I changed the C for B. Now instead of the cat saw the dog, we have the bat saw the dog. Can you see that now this triplet codon changed, and perhaps instead of coding for the Y amino acid, is going to code for another type of amino acid, which can change the whole protein structure. In our second example, instead of the cat saw the dog, you can see that I changed the letter in the last triplet codon. And now we have the cat saw the hog. Here, again, the change in one letter, known as point mutation, can cause, for instance, the change in the protein. Okay? In the last example, let's say that changing W for T, instead of saying saw, we say the cat sat. Here, we are adding a stop codon. And in this stop codon, the translation of the protein, which would go all the way to the end, now stops at this point. Instead of having a protein, let's say with 4 subunits, you have, for instance, a protein with only 2 subunits, and the third subunit looks weird because you stopped the synthesis before it actually terminated. So see that we changed the reading of that sequence. Now let's look at the frameshift mutation. Let's look first when there is the loss of one letter. So here I am showing that the cat saw the dog. Now we're losing the C of that phrase. And what we have now is the ats. Look at this. And because of the loss of the C, now the sequence shifted backwards, sort of, okay? So now you can see that the codons are completely different from our initial example. Perhaps now that the codons are completely different, the amino acids can also be different. So again, this mutation is changing the final protein. The other example is when there is a gain of one letter. And here you can see that in between the C and A, we inserted a letter M. And now the frame or the sequence shifted forwardly. And you can see that The codons also changed. The triplet codons also changed, which means that the amino acids that they code will also change, resulting in a different protein coded. The mutations can also be classified according to the effects on the function. And we probably talked about this throughout these classes. I am pretty sure I gave you some examples of these mutations already. The first one is the loss of function mutation. The loss of function mutation is when there is a reduction or there is an elimination of the gene, of the function of the gene product. The null mutation is when there is a complete loss of function. A recessive mutation is when the mutation is related to a recessive gene, which most of the mutations will cause loss of function as well. A dominant negative mutation is when the mutation is regarded to the dominant gene. And this dominant gene, when it is mutated, will produce a inactive gene, which also changes the final protein, changing the final function. We can also classify these mutations by the phenotypic effects. They can be visible when the alterations in that gene sequence change some visible morphological trait, meaning that you look at the organism and you can identify the organism is different from what you expected. We can also have a nutritional mutation, which is when that mutation change the nutritional characteristic. For instance, the ability of synthesizing a certain amino acid, or the ability of absorbing a certain nutrient, the ability of breaking down starch, something like that. The biochemical mutation is when the protein changes its function. For instance, the example I am giving you here is when the hemoglobin, which is the protein that carries the oxygen, is mutated, which causes this deficiency known as the sickle cell anemia. And in this sickle cell anemia, the cells can no longer transport the oxygen. Therefore, the biochemical pathway related to the transport of the oxygen is no longer feasible. So changed that biochemical reaction per se. We also have behavioral, regulatory, lethal, and conditional mutations. But the point here of this slide is to let you know that we can classify those mutations when the phenotype is changed, either in the external appearance, and that would be a visible mutation, or in some biochemical behavioral aspect of that organism. Mutations can also be classified according to their location. If the mutation happens in any cell except the germ cells, we call them somatic mutations. So for instance, if the mutation happened to the red blood cells, like in the sickle cell anemia, or happened in the skin cells, or happened in the cells of your stomach, those are all somatic cells because remember, they have 2n amount of chromosomes. Germ cells, again, only have n amount of chromosomes. Somatic mutations are usually not heritable, meaning that they won't pass to another generation. Most of the mutations will happen still in the germ cells, is still in the gametes. they will be called germline mutations. Those germline mutations can be inherited and will be inherited because the germ cells or the gametes is what makes up a new individual. Therefore, if the gamete is altered, the individual the gamete produces will also be altered. We also have autosomal mutations, which is when the mutations occur in genes located in the autosome chromosomes, which is, in the case of the humans, any of the 22 pairs except the sexual chromosomes. And the opposite of that is the X-linked and Y-linked mutations. We also dedicated one class to talk about X-linked inheritance. Here, the mutations are associated with the chromosomes X and Y, only those two chromosomes. Therefore, if mutations are associated with any other chromosomes that are not the sexual chromosomes, they are called autosomal mutations. Remember that many of the examples I used in other classes, we use the terms autosomal recessive, for instance, because that mutation was happened in a chromosome that was not the sex chromosome. In nature, mutations can occur spontaneously or induced. And we are going to briefly, in the next slide, talk about those two forms of mutation, just so you know the differences between them and what can cause them. Spontaneous mutations, as the name suggests, are those changes in the sequence of the nucleotides of that gene that will occur in a natural form. Okay, so there is no exogenous factor altering that nucleotide sequence. Spontaneous mutations often occur because of fail that happens to the DNA polymerase, which is the main enzyme involved with the DNA replication. Other enzymes can also fail during that replication. However, the most common is definitely the errors associated with the DNA polymerase. Okay? Induced mutations, as the name suggests, are those mutations that will occur due to the presence of exogenous factors that are outside of the body, that are influenced by things in the environment, such as radiation or UV light. Those factors can be, for instance, natural or artificial, okay? And you probably have heard of many chemicals, for instance, that might be bad for your health because they do cause mutations. They do change the nucleotide sequence of your genes. Here, I want to give a more specific attention to the spontaneous mutations that can occur during the replication process. The most common things that can occur during that replication process is when the bases are modified. So what we are gonna look in the next few slices is how the DNA polymerase can fail, can commit mistakes, and how those bases can be modified. So those errors, let's think about the DNA polymerase, and those bases, we're going to think about the nitrogenous bases that we have been discussing. First, let's talk about the DNA polymerase. So, one of the things that can happen with this DNA polymerase is that it can insert incorrect nucleotides. So if you think about the DNA, and I'm always going to represent just one strand, but always think that the DNA has two strands, right? And let's say that the normal sequence here would be A, T, and C. The DNA polymerase sometimes can, instead of putting an adenine here, puts another base, such as guanine. Here, some of these changes caused by mistakes with the DNA polymerase will end up not causing big changes in the phenotype. They will end up being a mutation that does not result in a bad phenotype or in an apparent phenotype. Okay? But this happens because most of the times the DNA polymerase will misincorporate those nucleotides in the sequence. And if those misincorporated nucleotides are included in this sequence, then they will persist after replication, right? Because now the new letter is the one that will be present in the sequence, and that will be passed down to the replication process. If those errors are not detected and are not corrected, you can definitely lead to mutations. But like I said, the DNA polymerase often does insert incorrect nucleotides, and those mutations, those changes are not really detrimental to the organism. Okay? If I am not mistaken, I believe that the DNA polymerase inserts a wrong nucleotide every 100,000 or probably 10,000 nucleotides or even more. So it is about, to every 100,000 nucleotides that it puts correctly, one will be put incorrectly. But again, like I said, maybe this new codon will not change the amino acid sequence. So that mutation will not actually cause a difference in the phenotype. Here, what can also happen is the bases, those nitrogenous bases, assuming different forms that we call tautomers. And we have a specific slide to talk about what are these tautomers and how they affect the replication process. Another process that can happen is the replication slippage. Here, let's think that the DNA polymerase literally slips, literally jumps that sequence, okay? So here in the replication slippage, we can define as a error that will occur during the DNA replication when that DNA polymerase dissociates temporarily from the template strand. Remember that we have the lagging strand and the template strand. So here the DNA polymerase temporarily dissociates from that template strand during the replication and then reattaches. But when it reattaches incorrectly. Here, this slippage of the DNA polymerase often happens in these regions that we call hotspot. Those hotspots are regions in the DNA with repetitive sequences. And here, these regions with repetitive sequences are often target of this, I would call this mistake caused by, or this fail in the DNA polymerase. Okay? Here, because of this replication slippage, we can have some of those frameshift mutations. Remember that the frameshift mutations, what happens is that the sequence is shifted either backwards or forward, right? And that can change the triplet codons, which then changes the protein or changes the amino acid that can change the protein. One common example or genetic disorder that is caused due to the replication slippage of the DNA polymerase is the Huntington disease. And we've talked about this condition over and over in our classes. In the previous slide, I mentioned about the tautomeric format of the nitrogenous bases. These tautomers are actually alternative forms of the nitrogenous bases. So the purines and the pyrimidines, which are the two classes of the nitrogenous bases, they exist in this tautomeric form, which is a, I don't want to get too involved with chemistry here, but they undergo through a transient structural isomerization. So let's think that it is, by isomerization, let's think of that base, it is similar, but some of the atoms that compose that nitrogenous base is changed transiently, okay? And that occur, this transient structural isomerization of the nitrogenous base usually occurs due to the repositioning of the protons in those atoms, okay? Which will lead then to this tautomeric form. We have two specific tautomeric forms, the keto-enol, and I'm gonna write it here, but I will also show in the other slide. Keto-enol and the amino okay, those are two tautomers. Those are two alternative chemical forms of those nitrogenous bases. Here, if those bases assume this tautomeric form, they can actually lead to a change in the strand of DNA. And if you change the sequence in that strand of DNA, then sometimes you can have bases that are non-complementary to each other. So if you think of the DNA as a double strand like this, and then one base is pairing, is complementary to the other base in the other strand, if you have the tautomerization of this base, it might be the case that they are no longer complementary to each other. And because of this non-complementation between those bases, you can have a mismatch base pairing, which can cause point mutations. And those point mutations can lead to permanent changes, okay, in the DNA structure. This picture is just exemplifying, it's just an example of what is that tautomerization. So normally, as you might remember, adenine pairs with thymine. Thymine has the chemical group keto, and adenine has the chemical group amino. And they bound with each other, as you can see in this first example. However, if the thymine assumes its enol form, the enol form is a tautomer. It looks similar, but is not the same. It is similar, but not identical. I think that's what my professor would say. Here, as you can see, you have a change in the functional group of that thymine, of that nitrogenous base. Because now you have a change in the group of the thymine, you have now a guanine connecting to the thymine, which was not possible, but now it is, because this version of thymine can allow for this pairing, for this matching. And this is obviously a mismatching. Just so you remember, A pairs with T and C pairs with G. The other example is the cytosine and the guanine. Normally, cytosine carries a amino group and the guanine carries the keto group, okay? However, in the tautomeric format, the cytosine now can carry the imino group. It's no longer the amino group, it's the imino. And in this case, the cytosine that was once paired with the guanine now pairs with another adenine. So you can see that in this top part, we would have the normal pairing of the bases. But at the bottom part of my figure, I am showing you that T is pairing with G and C is pairing with A. And that happens because T and C assumed their tautomeric format, okay? Which is not common. It is a type of mutation that happens to the base pair, not to the DNA polymerase, okay? Let's look at the effect of this tautomerization to the DNA. Here I am showing you at the top, I am showing you a DNA with both strands. You can see that A pairs with T, T pairs with A, G pairs with C, and C pairs with G. Okay? When the DNA replicates, we need to open this molecule, right? And then you have two strands of DNA. One is the template, the other one is the lagging strand. Here you can see that in this strand there is a tautomeric shift, meaning that once the adenine that was carrying the amino group now carries the imino form. Now, because of that, because of this tautomeric shift, when there is the first round of replication, the A no longer pairs with T. It now pairs with C. So you can see that this strand of DNA is completely different from this where no tautomeric shift occurred in the nitrogenous bases. Here you have a DNA with no mutation, and then on the other side you have a DNA mutated, because now you have the C in the position where it was supposed to be T. And then as the replication goes through multiple rounds, that change persists throughout those replications. And as you can imagine, the codon here is completely different from the original codon, meaning that the amino acid created in this new codon can be completely different from this codon right there, which can then change the phenotype. Two other events that can happen with the nitrogenous bases are known as depurination and deamination. As you can imagine, the word D in both events means lack of. Okay? So depurination is when you have a loss of a nitrogenous basis that is usually a purine. And in the purine, we have the guanine or adenine. The loss, the lack of that purine base in that DNA strand leads to a apurinic site, which means it's a space. It is a site without a purine base. A deamination, as you can imagine, is the loss, the change of the amino group in the molecules of cytosine or adenine. In this case, the amino group is converted to the keto group. Remember, the keto group is only present in the thymine and guanine. And the amino group is present in the cytosine and adenine. Okay? Here, when you have this deamination happening to the cytosine, what happens is that the cytosine now is converted to a molecule of uracil. And if it happens to the adenine, adenine is converted to a molecule of I am very sorry about pronouncing this structure right there. Here, what happens now is that the A and T, the adenine and thymine pairing, is converted to a guanine and cytosine pairing. Okay, so as you can see, there can be a change in the base pair in those two positions of the DNA strand. Okay, this is an example. So here, the cytosine molecule has the amino group, and this amino group, if it is changed to a keto group, now you convert this molecule to a uracil molecule, and the uracil molecule, as we know, binds to an adenine molecule or an adenine nitrogenous basis. Okay? The same thing can happen with the adenine molecule that also has a amino group. The adenine can also go through deamination, and in this case you have the formation of this different nitrogenous base, which now binds to cytosine, which we also know it was not possible because adenine only binds to thymine. Okay? As I presented before, besides of the mutations having the chance of occurring naturally or spontaneously by mistakes with the DNA polymerase or alterations in the nucleotides, those mutations can also be induced. And those components that cause those mutations are usually chemical components or radiation. And we are going to look at how those chemical components and how radiation can change or can damage the DNA. We call those components mutagens. So mutagens are all those components that induce mutations in the DNA. Every single day, we are exposed to different mutagens. They can be fungal toxins, medical x-rays, UV lights, or even chemicals in tobacco or chemicals in the food that we eat. Those mutagens sometimes can work as a base analogs, meaning that those mutagens, they can replace the nitrogenous bases. They can replace those nucleotides. They can substitute the purines and the pyrimidines when the DNA is being replicated and synthesized. When that occurs, you increase the chance of the tautomeric shifts. Remember, tautomeric shifts or happens when a certain molecule can also be found in its alternative version. But that alternative version sometimes gives the molecule a property that molecule did not have. Remember that when you have the tautomerization of the cytosine starts binding, for instance, with an adenine molecule. Okay? When this occurs, you can also increase the sensitivity to the UV light, the sensitivity of the DNA to UV light. And here I'm going to give you an example of the 5-bromacil, which behaves as a analog for the thymine. Okay? So here in this slide, I am showing you how the thymine is usually found in nature. However, when this component enters in contact with this with the DNA, it replaces the thymine. And sometimes this component, 5-Bu, can also be found in its tautomere format. And when it is in the tautomere format, what's happened is that the thymine, or this analog of thymine, now binds to guanine. Remember that thymine only binds to adenine. However, when you have this chemical component, now the thymine molecule can also bind to guanine due to those tautomeric shifts. There are 3 special agents that I also want to talk about. They are known as alkylating agents, intercalating agents, and adduct forming agents. Starting with the alkylating agents, they are mutagens that will donate the Aq group to a amino or the keto group of the nucleotides. Remember that the cytosine and the adenine, they have the amino group, and the thymine and the guanine, they have the keto group. That's just the chemical composition, okay, of these nucleotides. Alkylating agents can donate those Aq groups, and that changes the base pair affinity. So looking at this example, you have a molecule of guanine. The molecule of guanine always binds to cytosine. However, when you have this mutagen, when you have this alkylating agent, which is a mutagen, what happens is that now guanine binds to thymine. And again, that can alterate the DNA sequence. That can alterate the codons, the triplet of codons. Okay? We also have the intercalating agents. The intercalating agents are chemicals with dimensions and shapes that resemble a nucleotide. Because they resemble this nucleotide, they kind of get in between the DNA strand. And when they enter the DNA strand, that will cause a base pair distortions, right? Because you will cause that frame shift mutation, because you will push the nucleotides either backwards or forward. Okay? So because of that, you can cause those frame shift mutations. So remember, you have the molecule of DNA with the base pairs and the nucleotides over there. These intercalating agents would position themselves in between those nucleotides, which then would change the composition of those triplet codons. Okay? This also promotes a DNA unwinding. Because of these effects of the intercalating agents to the DNA, we can have effects this can affect the transcription, replication, and also the repair machinery of the cell. The last agent I want to talk to you is the adduct forming agents. The adduct forming agents, we have two that you probably have heard about. One is the acetaldehyde, and the other one is the heterocyclic amines. This first one is usually found in the tobacco smoke. It's usually found in a cigarette. So you probably have seen many campaigns, to stop smoking cigarettes because they can cause cancer. And that is because those cigarettes, they usually have this component, this mutagen component. The second component, the heterocyclic amines, they are found in meats when you cook them at higher temperatures. They are also mutagen agents, and them both, those two examples of adduct forming agents, they bind to the DNA, and when they bind to the DNA, they change the conformation of the DNA. By changing the conformation of the DNA, you can expose the DNA to enzymes that can degrade the DNA, okay? Or to any other agents that can affect the DNA. Another agent that can induce a mutation is the UV light. The UV light usually creates pyrimidine dimers, which means that the molecule of DNA, wherever the UV light hits in the molecule, will have a duplication in the pyrimidines, which then, as you can imagine, cause a change in the conformation of the DNA and also cause a change in the triplet codon. Here's an example of a thymine dimer. So if the UV light hits this normal molecule of DNA, this causes a duplication of the thymine, and now you cause that frame shift mutation, okay? Which shifted the DNA, which shifted the triplet codons. Another agent that can induce mutations is the radiation, the ionizing radiation. Radiations that can cause mutation to the DNA, you probably have heard about them. They can be, for instance, the X-ray, the gamma rays, and cosmic rays, or any other type of wavelengths that can change the DNA. These guys, this type of radiation, cause ionization of the molecules. And when it causes ionization of the molecules, it produces what we call free radicals. Now let's try to understand this process step by step. Free radicals we can understand them as highly reactive chemical molecules. And those reactive chemical molecules, they usually have one or more unpaired electrons. If that electron is unpaired, there is no pair, there is no matching for that electron, that makes the molecule unstable. And if the molecule is unstable, that molecule interacts with other molecules, such as the DNA. Usually, this radiation can penetrate the cells, so that high energy radiation, such as X-ray, gamma rays, they can enter the cell, and because they are carrying a lot of energy, they ionize the molecules, literally stripping the electrons of those molecules, creating that instability with that molecule. Those unstable molecules, they are transformed into free radicals. So that radiation interact with cellular molecules, such as water, for instance. So water, sometimes if it interacts with radiation, can produce free radicals, such as this one. And we are going to represent as a dot to represent the unpaired electron. This is a free radical known as hydroxy radical. Okay? This can also lead to the production of the O2, and I think there is a minus and a plus sign here. So this is what we would call the superoxide radicals. Both of these radicals that are formed when radiation interacts with the molecules of water, they attack the DNA. But not only the DNA, they also attack other cellular components. Okay? But anyways, by attacking the DNA, it can damage the DNA by, for instance, altering the nucleotide bases. Okay? This type of radiation can also break the phosphodiester bonds. Remember that the DNA has let me just represent here, has the sugar phosphate backbone, right? Which is this portion right here, where you find sugar and phosphate. So between each one of those molecules, you have this phosphodiester bonds. Those free radicals can interact with those bonds and then cause the cleavage, can literally break those bonds, which causes then the DNA to be single strand or cause a double strand break, for instance. As a result of these free radicals interacting with the DNA, you can have, for instance, deletions, which is when the genetic material is lost. You can also have translocation, and by translocation, let's understand as the rearrangement of the chromosomes, the rearrangement of the genes in that chromosome. And you can also have, like I said, fragmentation. That's when the DNA breaks apart, literally breaks apart. That's why those radiations are very concerning, and they can induce mutation, okay? Because they literally produce free radicals, and those free radicals can interact with the molecule of DNA, causing deletion, translocations, or fragmentation. I want to now give you some examples of some disease that are caused due to mutations to the DNA. But before we talk about those examples, let's just remind of this concept. And I believe I've talked about them, or I gave you examples about them throughout the semester already. So most of the human genetic disease are polygenic, meaning that they are caused by variations in several genes. Okay, so many of the genetic disease that we observe in a population are caused by changes, mutations in several genes. However, I believe that in some of the examples I gave to you, for instance, the Huntington disease or the sickle cell anemia, some of these genetic disease are caused by a mutation in one single gene. One exclusive gene is associated with that mutation. Okay? So in this table, I am giving you some examples of genetic disorders that are caused by mutations in one single gene. Just remember that here I am showing you the type of mutations, and here are the disorders. Missense mutation is when the mutation results in a new triplet code, okay? And that new triplet code gives you a different amino acid. A nonsense mutation is when that triplet code is actually a stop codon, and that causes the termination of the translation earlier, prematurely. Okay? Insertion is when there is an insertion of the nucleotide in that sequence, which can cause some frame shift mutation. Then we also have deletion, which is literally when that nucleotide is deleted from the gene. And the last one is expansion, which is also associated with the frame shift mutation, okay? Because you have insertion or multiple insertions in that genes. Those disorders can be achondroplasia. Achondroplasia, like the name suggests, and let me get rid of this annotation. So achondroplasia, like the name suggests, A means lack. Chondro means cartilage. Okay? So here, this genetic disorder causes dwarfism, for instance, because there is a impaired bone growth. So in this case, the bone doesn't actually develop because of a mutation in this FGFR3 gene. A mutation in this gene, a missense mutation, causes a abnormal development of the cartilage. And cartilage is a component that surrounds, very basically surrounds the bones. Okay? This will cause a shorten of the limbs, for instance. The other disorder, genetic disorder, is the Marfan syndrome. This is a nonsense mutation, meaning that the new triplet codon is actually a stop codon, meaning that the translation terminates earlier, okay? In this case, in the Marfan syndrome, we have a genetic disorder that affects the connective tissue. And here, this protein, fibrillin 1, is no longer produced. Here, this protein is very important for the elastic fibers that exist in the connective tissues. Without the proper fibers, these connective tissues, they don't actually execute their function. And where do we find connective tissues? We find connective tissues, for instance, in the bones, in your eyes, around your heart, in your heart. We can also find in the blood vessels. So there is connective tissue all over the body. So without this protein, there is no elastic fibers, and this can lead to many types of complications, cardiovascular complications, complications in articulations, and can cause, for instance, heart issues, blood vessel issues, okay? But this is a type of a autosomal dominant inheritance disease, autosomal dominant type of disease, okay? Meaning that you need a dominant allele of that gene to cause the mutation. The other one that I have here is the familial hypercholesterolemia. I know this is a big word, but let's break it down. Hyper means higher, more. This cholesterolemia means cholesterol. So as you can imagine, this genetic disorder is responsible for causing high cholesterol. And here specifically the cholesterol LDL. The LDL cholesterol is the bad cholesterol. Is that cholesterol that when you go to the doctor, if they find that it's high, they will say, oh, reduce the amount of fat that you are eating, okay? This, when the LDL cholesterol is increased, it can also increase the chances of heart disease, for instance, or a very common disease caused by high LDL is atherosclerosis, which is the accumulation of fat in the artery, okay? And this specific condition, genetic condition, is caused by a autosomal dominant inheritance as well. The other one is the cystic fibrosis, which is due to a deletion, right, in the gene. This, you probably researched this in your clinic case study. This is a recessive genetic disorder, okay, which is caused by mutations to the gene CFTR. And this will cause the accumulation, the thickening of the mucus in your airways, in the ducts connecting your pancreas to your small intestine, okay? And this, for instance, can cause respiratory issues, can cause issues with absorption of nutrients, okay? You'll probably recall of this genetic disease when you did, again, your clinical case study. The last disease here is the Huntington disease, which is caused by a mutation that is actually an expansion of the gene sequence. Here, the gene sequence is the HTT gene. And here you have an expansion, you have an addition of many of the same nucleotides. Okay, and in this case, the nucleotides are CAG. In that gene, in that specific gene, HTT gene, you have about 40 repeats of this triplet in that sequence, which, as you can imagine, cause a change in the whole gene, right? A very common symptom of the Huntington disease, because it is a neurodegenerative, I'm sorry about mispronouncing this word, is a neurodegeneration. Is a neurodegeneration, will cause, for instance, motor issues, cognitive issues, psychiatry issues as well. But the main idea here is just to give you an example of these mutations, some of them that we've talked over and over throughout the classes, such as cystic fibrosis and Huntington disease, but so you can correlate the missense, nonsense, silent mutation, expansion, insertion, deletion that we've been talking about throughout this class. In the same way that mutations can change the DNA, organisms also have a system of enzymes that can help repairing, readjusting, correcting those mutations caused in the DNA. So the repair systems that exist either in prokaryotes or eukaryotes, they will always counteract they will always try to correct those damages, those mutations in the DNA, either if those mutations are spontaneously, if they are spontaneous or induced. Okay, this system, and I'm gonna discuss about some examples, they help maintaining the integrity of the DNA, making sure that those genetic diseases are prevented. Okay, and I know I've listed cancer here, but cancer is also can be a type of genetic disease. Okay, the first system that I want to discuss with you is the post-replication repair system. As the name suggests, this post-replication repair system responds after the damaged DNA escaped the repair and the replication has failed. I am going to show you a picture because that's going to be easier to understand. But here, the main protein that helps detecting this, not only detecting but also like conducting the repair, is known as the RAC-A protein. So the RAC-A protein will govern, is the main key here in this repair system. So let me show you this figure because I think it will be easier to understand what I am saying. So here you can see that the DNA is opening. So this would be our replication fork. And you can see that there is a lesion right here in this DNA. And let's assume, because this is a thymine dimer, let's assume that this lesion was caused by UV radiation, for instance. Okay? In the other region, in the other DNA, in the other strand of that DNA, you have the complementary region. And as you can imagine, if you have two thymines, you then also have two adenines. Okay? Here, what happens is that if replication is occurring, the DNA polymerase will skip the lesion here. So as you can see, the replication is skipped over this region where you had the lesion and continues. Okay? But what happens is that the enzyme RAC-A recognizes this region that has a lesion and then drives the following process. The undamaged complementary region of the DNA will donate the complementary nucleotides to the new strand of DNA. So see that here we have a gap, and that gap needs to be filled. The gap will be filled with the two adenines coming from the complementary strand. So they are moved up to match the thymine dimer, and the new gap that you have there is then filled by the DNA polymerase and by the DNA ligase. Remember that the DNA polymerase will bring the nucleotides and insert the nucleotides in this gap, and the ligase will make sure that everyone is bound together, is linked together. Okay? So it is called post-replication because the DNA polymerase skips the site of lesion, and then you have other enzymes that detect the site of lesion, and then there is a recombination process from which is the complementary base pairs in the complementary strand of DNA is donated to that newly formed DNA strand. And then this new gap in the complementary DNA strand is corrected by the DNA polymerase and the DNA ligase. The other system, the other repair system I want to talk to you about is the base and nucleotide excision repair. Excision means removal, cut, okay? So here there are two types of excisions that can happen. One is the base excision repair, and the other one is the nucleotide excision repair. Let's look at the base excision repair first. I'm going to show you also a picture because that will make things a little bit easier. But just so you understand, there are two main enzymes in this base excision repair system. The first one is the DNA glycosylase. This enzyme will recognize the base that was changed, will recognize the lesion, will recognize the site of the mutation. The other enzyme is the AP endonuclease, and the endonuclease will cut the phosphodiester bonds in the sugar phosphate backbone. Okay? Then you also have the DNA polymerase and ligase filling the gap. But let's look at the picture to make more sense. Here you have the DNA with the two strands, right? And here you can see that by mistake there is a uracil where it should not be, because if you have G, the only possible pair should be C. But as you can see, there is a mutation there. There is a change in the base pair. Okay? What happens is that the DNA glycosylase will recognize this mutation. And then it will call its partner, the AP endonuclease, and the AP endonuclease will cut. And as you can see here, there is a gap now. We'll cut the site of the mutation, okay? Removing, it literally excises. I think that's the right verbiage, right? It literally cuts that nucleotide from that position. And here, what happens then is the DNA polymerase comes and bring the correct base, and then the DNA ligase will bound that nitrogenous base back in the backbone. Okay? So again, the base excision repair, base means that we are removing, we are cutting off or cutting out the base pair where the mismatch, where the lesion, where the mutation occurred. Two enzymes are important. They are the DNA glycosylase and the AP endonuclease. DNA glycosylase recognizes the site of lesion. AP endonuclease cuts the size of lesion. Then you have DNA polymerase and ligase to repair that site. The other system is the nucleotide excision repair. And again, I'm going to go straight forward to the figure because it will be easier for you to understand. In this type of repair system, we have the product of the gene known as UVR. UVR because this mutation is usually associated with the UV light. Okay? So we give the gene that is involved to the repair of the lesion the name of UVR gene. This is a figure to kind of guide you through the process. So here you have a damage to the DNA that, for instance, can be caused by UV light. UV light. Here, what happens is that you have a nuclease that will cut a big portion now of this DNA. Remember that in the base excision repair system, we only had the change of one single base. But here, we are actually removing a big chunk of this sequence, okay? And the nuclease is the enzyme that serves as a scissor that cuts this site of lesion. Then you have the DNA polymerase 1 bringing the new nucleotides, the correct nucleotides to the gap, and the DNA ligase sealing the gap between those pairs. Now, remember that I told you that those free radicals, and I think I gave you the example of the hydroxyl and the superoxide radicals, free radicals that can be formed from the ionization of the water, right, through the radiation. So here, they can cause these breaks in the double-strand DNA, right? So double-strand meaning that you have the DNA, and the DNA has two strands, okay? And these free radicals can affect and break, literally break, the phosphodiester bonds in the sugar phosphate backbone of that DNA, okay? We have two systems, two pathways to fix this type of mutation. They are known as the homologous recombination repair and the non-homologous end joining. Let's talk about first of the homologous recombination repair. And I'm going to show you a picture because I think it will make more sense when I show you the picture. I just wanted to call your attention that this type of repair pathway occurs in the late S phase or early G2 phase of the cell cycle. If we represent the cell cycle here, remember that it starts with the G1 phase, then you have the S or synthesis phase, and then you have the G2 phase, okay? This process of homologous recombination repair will happen somewhere at the end of the S phase or at the beginning of the G2 phase. Why is that the case? Because in this type of repair system, we need a sister chromatid. And the sister chromatid will serve as a template for the DNA to be fixed. And we need here the DNA to be duplicated just so we can have a sister chromatid. Before the DNA is duplicated, there is no sister chromatid, right? Because there is only one arm of that chromosome, right? After the DNA is duplicated, you have two arms of that chromosome. In this case, one portion of the sister chromatid will serve as a template to repair the region where the mutation occurred. Now let's look at a picture to make more sense of what we are saying. Let's imagine here that you have your double-strand DNA that was broken because of a free radical. Here, as you can see, there is a gap in this double strand because, like the name suggests, the break is putting those strands apart. What happens is that this DNA will use the DNA in the sister chromatid of the sister chromatid as a template to manufacture the missing sequence. So here you have the DNA where the mutation occurred. And here you have the sister chromatid. Okay? Now, what happens is that the 3-line ending matches with the complementary region in the sister chromatid. Okay? And the 3-line ending of both strands, as you can see, of the template and the leading and the lagging leading and the lagging strands of DNA. Okay, they will find their complementary region in the sister chromatid, and that will serve as a template for the DNA polymerase to come here and insert the nucleotides. Okay, nucleotides that are missing. And as you can imagine, you have the DNA ligase making up the bonds, making up those phosphodiester bonds that were broken. And at the end, you are left with a chromosome with 2 arms. So here we are representing the chromosome with 2 arms. Okay? At the beginning, you only had 1 arm. But as the cycle progresses from G1 to S phase, now you have a second arm. So this is the first, this is the second. What happens is that if this breaks, then the portion that it was broken can use the sister chromatid here as a template to recompose, to manufacture that missing portion of the DNA. And then at the end, you are left with a normal 2-armed, let's put this way, chromosome. The other system is the non-homologous end joining pathway. This is activated at the G1 phase before replication begins, before the S phase, before the synthesis phase. Like the other system, it also repairs the breaks that happens in the double strand of DNA. Here, the proteins very simply, much more simple than what happened for the homologous recombination repair pathway. Here, the proteins will bind the free ends and bring them back together. Okay? Simply like that. So imagine that you had this double-strand DNA that was broken. Okay? What's going to happen is that some proteins, such as the DNA ligase, will bring those portions, will bring those sugar phosphate backbones back together. Okay? Again, much more simple than the homologous recombination repair system. All right? In the last portion of this lecture, I want to talk to you about these transposable elements, which we can also call transposons. These transposable elements, even though we don't have or we don't know yet their precise function for the cell or for the human or for the organism's genome, we do know that they contribute somehow to creating mutations and also to evolution, right? Mutation is a evolutionary force. So these transposable elements are also known as jumping genes. And they are known as jumping genes because they literally move around in the genome. So they literally jump from one site to another region. Okay? These transposons or transposable elements are DNA sequences that move within and between the chromosomes. So within would be in the same DNA sequence or in the same DNA, and between would be to a sister chromatid or to another homologous chromosomes. And when they jump from their original location to another location in the genome, they insert themselves in that new location. And by inserting themselves in that new location, you can expect that they change the sequence of nucleotides. They change the sequence of bases within that region that they are now inserted. And that's why they promote mutations and might contribute to the evolution of the organisms, okay? But if they are actually causing, if they are actually coding for a protein involved in a important biochemical pathway, or if they are involved in any other bodily function, we still don't know. There are two types of transposable elements, okay? One of them is the DNA transposons, and the other one is the retrotransposons. We're just gonna take a look at how they look, their composition, but we are not gonna go into details about them. I just wanted to give you a brief introduction about this, because if you take future courses and higher level courses, for instance in molecular genetics or molecular biology, you will probably listen about these transposons again. So the first one is the DNA transposon. So the DNA transposons are those jumping genes. So they move from one location to another, but they do not use the RNA as an intermediate stage. Okay? The retrotransposon, they do use RNA, but the DNA transposons do not. The DNA transposons, and I am going to show you a figure of what they look like, but they have these ITRs repetitions. Those ITRs repetitions are sequences named as inverted terminal repeats, and they are located at the end of the transposon, and they delimitate, they flank the whole transposon. Okay? These ITR regions, they are recognized by the transpoase. The transpoase is an enzyme that cleaves, that cuts the transposon. And by cutting the transposon now is able to move to another region. Okay? So transpoase is an enzyme that cuts, that recognizes these ITR regions in the transposon. Okay? The transposon also has what we call ORF region, that stands for open reading frames. The open reading frames is a region in the transposon that codes for the enzyme transpoase. So it is a sequence of nucleotides within the transposon that has the information to produce the enzyme that cleaves the transposon, essentially. The enzyme is required for the movement of the transposable element to another region, in and out of the genome. So this is how the transposon actually looks like. So here I am representing the sequence of this transposon. As you can imagine, this is our chromosome, and what I am showing you is what are the elements of these transposons, the DNA transposons. So the transposon here is shown in red, and they are flanked by these DR regions. The DR regions shown in blue are direct repeat regions. No specific function related to them so far. So within this transposon, we have the ORF region that codes for the transpoase. which is the enzyme that cleaves or that recognizes the ITRs, the repeats, the inverted terminal repeats, and cleaves the transposon out of the genome. When the transposon is cleaved out of the genome, it floats, it jumps to another chromosome or to another space within that genome and inserts itself in that space. Okay? Which, as you can see, causes one of those frame shift mutations, okay? Because it shifts the sequence, the original sequence that was in that chromosome. The other type of transposable element is the retrotransposons. The retrotransposons receive that name because it uses the RNA as intermediate. Retro, and you probably have heard about retrovirus, they use the reverse transcriptase, which is an enzyme that will make DNA out of the DNA. So here, the transposon is also a mobile genetic element, a jumping gene, that will move within the genome. But here, they are going to use the RNA and make DNA out of that RNA using the reverse transcriptase, the enzyme reverse transcriptase. Okay? Once that new DNA is fabricated or is manufactured, then it is inserted into a new genomic location. Similarly, these type of transposable elements also contribute to mutations, therefore also genome evolution. Okay? Like I said, they resemble the retroviruses because they use the RNA or the reverse transcriptase. Okay? So they make DNA out of DNA. Normally, according to the central dogma, you would have DNA making RNA, correct? And that process is called transcription. However, in these retrotransposons, they will use RNA to make DNA, and the DNA will insert in the genome. Okay, it will be inserted into the genome. So here, the enzyme used, or the process the process of making DNA from the RNA is known as reverse transcription. Okay? This is very common in some viruses. There are two types of retrotransposons. We're not going to look into them because that's way too much detail, but they are differentiated by this LTR element. The LTR element is just as the same as the ITR elements in the DNA transposons. They flank the end of the retrotransposons. And you have the long terminal repeat and the non-long terminal repeat. Okay? That's the only difference between them, and this is a picture of what they look like. Similar as the DNA transposons, they are also flanked by the direct repeats at the end, and they have this LTR region, okay? Right there. In the middle, you also have the ORF region. But here, the ORF region, this open reading frames region in the retrotransposon, will code for two enzymes. One is the integrase and the other one is the reverse transcriptase. The integrase will help, like the name suggests, integrating that transposon into the DNA. The reverse transcriptase, like I said, will help making the DNA out of the RNA. Okay, so the reverse transcriptase will follow the opposite process established by the central dogma. Okay? So this open reading frames, which also exist in the DNA transposon, is different here because in the DNA transposon, we were making the transpoase, which cuts the transposon from the DNA. But here, we're making two things, the integrase and the reverse transcriptase, which are both enzymes used, one, to integrate that transposon into the DNA, and two, to make DNA out of RNA. Okay? That's the maximum that you should know about this. All right? So the transposons are very important for mutation, very important in the sense that they can cause mutation because they spam the DNA where they are inserted, in the region that they are inserted. So because of that, they can cause what we call translation disruptions, okay? Because they move the DNA around when they insert into the new sequence of DNA. And that, of course, has effects on the gene expression, okay? So you change the whole configuration, I would say, of that gene because you're inserting new base pairs within that region, okay? And then because of that, it's proposed that these transposable elements, they contribute to the evolution of the species by altering those genes, by altering the gene product, okay, that was originally given by that gene. It is estimated that probably around 0.2% of the mutations in the human DNA might be due to the insertion of these transposable elements in those genomic regions. All right. So even though they don't have a specific mutation, we believe that they contribute to the evolution because they alter they change the composition of the genome because they jump from one site to the other. And I believe that that's all that we have for this class. I understand that we've talked about a lot of different mechanisms, but if you can pack them in, what's causing mutation and what is repairing the mutation, then I think you are good to understand what this class is about. The fact that we discussed a little bit about transposable elements, it's not as relevant as understanding the ways the DNA can be damaged and the ways the DNA can be repaired. Okay? Good luck when you go back and review this class.